So here's a typical retrovirus. These are the kind that Rouse identified, Rouse sarcoma virus and related viruses. On the left is an electron micrograph. So these are roughly spherical particles. They have a lipid envelope, and you can see there's a dark core in the middle. This is where the genetic information is. This is, in fact, the capsid. You can't see it very well on here, but there are glycoproteins. So on the right is the schematic. Starting from the outside, the glycoproteins embedded in the membrane. The glycoprotein is composed of two parts called surface and transmembrane. I'll often refer to it just as the viral glycoprotein. We have a lipid envelope. We have a protein, a matrix protein below it, which gives the envelope a little bit of strength and integrity. And then below that is the viral capsid, and that contains the nucleic acid of the virus. This is not an icosahedral capsid. It has some kind of hexagonal symmetry, but it's not built by icosahedral symmetry. The capsid contains the RNA genome, which is shown in green here. The RNA is complexed with a protein called nucleocapsid protein, and then there are a variety of other proteins included. There's a protease, a viral protease. There's an integrase and a reverse transcriptase, all within the capsid. All right, so that's our simple retrovirus. You'll see later HIV is a bit more complex than this. So here's the genetic coding strategy of these viruses, if you will. On the top, we're looking at what we call proviral DNA. So this is the viral genome converted to DNA as it is integrated in the host. And you'll see it's in blue because it's DNA. And there are two what we call long terminal repeats at either end, LTRs. We'll talk about what they are in a moment. And then you can see all the coding regions for the various viral proteins. So there are proteins here on the left that are used to build the core or the capsid. There are the enzymes in the middle, the reverse transcriptase and the integrase, which we will talk about in a bit. And then to the right are the proteins that make up the glycoprotein, the envelope glycoprotein, the surface and the transmembrane parts of that. The way this genome is expressed, it is always expressed when integrated into the host cell DNA. All right, so this proviral DNA, which by definition means integrated, is expressed. It's transcribed in the same way that mRNAs are made that we talked about last time by the host cell RNA polymerase II. And RNAs are produced. Here's a genomic RNA, which is full length. And this gives rise to structural precursors. It also gives rise to the reverse transcriptase. And then a spliced version of this gives rise to the envelope glycoprotein. So the envelope sequences are encoded here, but they can't be accessed in this message, so they're accessed by a spliced mRNA. So it's relatively simple. There aren't too many proteins, capsid, enzymes, and envelope glycoproteins. And as I said, the HIV genome is much more complicated than this, as we'll see later. All right, so how do these viruses replicate? Let's talk about the overall scheme. They bind to a receptor. There are many different kinds of retroviruses that we know of now, and we will talk about the human retroviruses that we know of later. They all bind to receptors, and they're all quite different. There are many, many different retrovirus receptors. The virus is taken up into the cell. Some of them fuse at the cell surface and release the capsid. Some of them come in by endocytosis. Either way, the capsid ends up in the cytoplasm. Okay, so now the membrane's gone. The capsid is there. You can see it's shown in a slightly permeabilized manner here. It seems to loosen up a bit as it comes in. But in that is the genome, the RNA, and all those enzymes and other proteins that we talked about. And this begins to synthesize DNA right away, right in the cytoplasm. And eventually it makes a double-stranded DNA, and then it goes into the nucleus. And we'll talk about these steps in a moment. The DNA gets into the nucleus where it integrates into the host cell DNA genome, which is shown right here. So the retroviral DNA is blue, and the host cell DNA is purple. From that position as a provirus, so this is what a provirus is, an integrated copy of the viral DNA in the host cell. If provirus is not this, it's not the RNA, it's this right here specifically. From that position, it is transcribed by host cell Pol II. There's a promoter near the 5' end we'll talk about briefly You make messenger RNAs. The host cell is making the messenger RNA. And these, of course, are exported. Some of them are exported without splicing to give rise to structural proteins and the reverse transcriptase and integrase. Some of these messenger RNAs are spliced and exported to make the glycoproteins for the envelope. And eventually, new genomes, glycoproteins, and structural components are assembled at the membrane, the plasma membrane, and a new particle buds off. We'll talk about this assembly process in more detail in a couple of lectures during assembly. What I find interesting about this is that the virus really doesn't replicate. It doesn't replicate its genome, right? The DNA doesn't replicate. Comes in and makes it RNA, it makes a DNA copy of the RNA, and then that integrates in the host cell. So there's no real DNA replication except when the host divides, of course, but the host is doing that. And then the virus makes messenger RNAs. So I don't really consider that replication. Some of these messenger RNAs end up getting into virions. So this is an example where the cell is doing everything for the virus. With the exception of that reverse transcription, the cell is making its DNA when it divides and making mRNAs. Now, reverse transcriptase is a very interesting enzyme. Let's talk about this a little bit. It's a primer-dependent enzyme. Primer can be either DNA or RNA. And the primer has to have a paired 3' hydroxy terminus. So here is an example of a template. So this is typically in the virus an RNA template, of course. And then we'll see what the primer is in the virion, but this in principle can be either DNA or RNA. And the 3' hydroxyl has to be free and base paired on the template right here, so that's what that means. The template can also be RNA or DNA, and you'll see where that comes up during the scheme of reverse transcription. Only deoxy NTPs are incorporated, not ribo NTPs, so it makes DNA. No matter what you give it, as a primer or template, it makes DNA. And of course, the right deoxy NTP is selected by base pairing with the template. So this is standard nucleic acid synthesis that we've already talked about here, with the exception, of course, that we're making DNA from an RNA template. Now, reverse transcriptase is quite an old enzyme. Once it was found in retroviruses and tumor viruses, then people started looking for it elsewhere to try and figure out where it had come from. So it turns out that there are some bacteria and archaea that have reverse transcriptase activity. The myxobacteria, E. coli, archaea, you can find RT activity in all of those bacteria. So we think that reverse transcriptase is quite an old enzyme. We think it evolved before the bacteria and the archaea and the eukaryotes separated here. So probably arose long before that, because they all have it. Right? It's a related enzyme, so it probably was in some precursor. It's thought that reverse transcriptase could be the bridge between the RNA world and the DNA world. You remember in the first session we talked about an ancient RNA world that we think existed, where all organisms were RNA-based. And that survived for a while. But you know, RNA is fragile. It's sensitive to nucleases. It can't get very long. You'll see that the biggest RNA viruses we know of, the genome is only about 25 kilobases long, much shorter than DNA genomes. So the idea was that although it worked in this early world, as soon as an enzyme arose that could make DNA, then DNA started to predominate. And we think that maybe RT was some enzyme that just arose by mutation from an RNA-dependent RNA polymerase, began making DNA genomes, and those thrived because they had a selective advantage. They could get bigger and they were more stable. This, of course, is all speculation, but it makes a lot of sense that RT is a very old enzyme. And of course, we had to find a way to convert the RNA world to a DNA one. So RT is quite old. Now, you remember a while ago we looked at the 4 classes of nucleic acid polymerases, and they're shown here again. This is the same slide that we looked at earlier. And these are showing you the RNA-dependent RNA polymerases the RNA-dependent DNA polymerases, reverse transcriptase, which we're talking about today, DNA polymerases, and then transcriptases, DNA-dependent RNA polymerases. And I remember I told you that they're all related. They have common motifs among all of them, these green areas which correlate with the thumb and the fingers and the palm domain. So RT fits in there as well. You can see it has the same domains and it has conservation. So probably these enzymes all arose from some common precursor. And these were some signatures for the other polymerases that I told you about. These don't apply to the reverse transcriptases. So reverse transcriptase has the ability to make a DNA copy of RNA. It's a primer-dependent enzyme. There is a second activity associated with reverse transcriptase that's essential for the retroviral life cycle, and that's RNase H. And this is an enzyme that cleaves RNA, so you can tell by the name, right? It's an RNase, only when it's in a duplex form. And that means double-stranded. It could be RNA-RNA or RNA-DNA. So it will cleave one strand of the duplex, and it cleaves endonucleolytically. It cleaves inside, not at the ends. It's not an exonuclease. It's an endonuclease. It cleaves on the interior, and it cleaves in this manner. So here is just a series of nucleosides linked together in a double-stranded molecule. And each one, of course, is linked by a phosphodiester bond, a single phosphate with two oxygens on either side. Here you can see it right here. So it's one base, oxygen, phosphate, oxygen. RNase H cleaves between the phosphate and the 5' terminal oxygen. So it makes short oligonucleotides with a 5' phosphate and a 3' hydroxyl. This enzyme basically is degrading the RNA as the enzyme is making a DNA copy of it. And we'll see how this happens in a moment. So that's RNase H. We do have structures at high resolution of many reverse transcriptases. This happens to be the reverse transcriptase of HIV-1 at the top here. You can see it has the motifs of a typical nucleic acid polymerase. It has a palm domain, and that's where the active site is. It has a fingers domain, a thumb. So it, again, looks like a right hand, as do the other polymerases. In this structure, which was actually solved with a bit of primer template in it, you can see that the nucleic acid lays across the palm. Here's the active site here with an incoming deoxy-NTP. So we have a template strand, which is being copied, of course, and a primer strand here as well. This cartoon at the bottom here makes it a little clearer. You can see the individual domains. This is the palm or the active site. And remember, the classic model for nucleic acid synthesis is the two-metal model. You need two metals that are coordinated by two aspartate residues. You can see them right here. Those are held in place to help coordinate the incoming triphosphate. And here, of course, the template is RNA. It's come in through this part of the enzyme. It's flowing across the active site. And then at the active site, DNTPs are added to the growing chain. So the DNA is in blue here. You can see it's moving across the surface of the enzyme, coming out here, this side, the opposite side where the RNA came in. And then down here is the RNase H active site. So the RNase H and the RT are part of the same molecule. The RNase H also requires two metals to do its thing. And this is degrading the RNA as it passes the active site. So the RNase H is taking away the RNA. It's not needed anymore. It served its purpose. It's a template for the synthesis of DNA. And that's coming out there. So DNA synthesis by reverse transcriptases is typically very slow. 4 hours to replicate a 9 kilobase genome. It's a lot of time, 9,000 nucleotides, and it makes a lot of mistakes. It makes one misincorporation for every 10,000 to 1 million nucleotides polymerized. So about an error per genome per replication cycle. It's quite error prone, and it doesn't have a correction mechanism to take away the errors. So this is why, in part, retroviruses are quite variable.